data analysis is over. So what are you going to do? How are you going to arrange them? When your examiner is going to read your thesis, it should tell, your analysis should be like telling a storytelling. So to do that logically, we need text. Some places we may use text, some places you may use tables, and some places you may use your figures. So what will you put is a question. So whether I'll put a table or I'll put a figure. So what is the question is based on your data. So what type of data you have. So this is basically the table to show what are different types of data. So we have two types of data. One is a categorical data and the numerical data. Categorical means qualitative, yes, no, male, female, smoker, non-smoker. Numerical data means the numbers, quantitative data. So in the categorical data, we have two groups, nominal and ordinal. So in the nominal, we say male, female, smoker, non-smoker. So it's mutually exclusive. Only these two you can write. You cannot write the third thing. Ordinal means there will be in some order, like disease severity, mild, moderate, in asthma, mild, moderate, severe. So that's also categorical. In the numerical data, we have again discrete data, number of visits, 1, 2, 3, 4, 5. Like that will come. Or it could be a continuous data, height in centimeters. We have 10 years old children, height may be different in different children. So you cannot write only one number, you have to give a range. So that is a continuous data. Because this table I'll come to the last, because based on your data, you have to use the what type of figure or table you are going to use. And remember that all your values in your study does not need a table, or you don't need to put a table or a figure for everything. Something very simple, maybe a text also. So for example, this is what we wrote in one of our PG's thesis. In children with arthritis, concentration of this particular protein was found to be 5 times high in the samples of patients with active disease compared to the patients who are in the remission. So we just wrote it's 5 times. There's nothing there to show in a table or a figure. So why do you want to use tables and graphs? One thing, when you put a table, a lot of things can be shown in the table. We can show the nice relationships, patterns, and trends very clearly when you use a table or a graph. And it also gives a visual perception of your data so that the reader or the examiner especially will know what is really happening. Sometimes when you put a lot of tables and figures, that also will confuse the readers. So what is a table? So see, whenever you write your thesis properly, it can be changed to a manuscript for submission in any journal very easily. So if you concentrate in your thesis writing by using the correct way or international standard of putting the tables and graphs, you can easily change that into a manuscript. Okay, so whenever you want to use a table, so this is the ideal table should be. This is one of the tables from our study. We published in Journal of Rheumatology a couple of years ago. So what we put is here we have two groups of people. So there are children with the SLE who had avascular necrosis. The another one is the children who have SLE but who don't have avascular necrosis. So AVN control. So what the table should be here? All the tables should have a legend. Remember that the table legend should come on top of the table. You cannot put a table legend on down. So you have to put a legend on the table legend. Then you have a column title. So every column has to have a title. You cannot leave it anything blank. Okay, you have to write characteristic features, parameters, lab parameters, radiological parameters. You have to write. So column title should not be left open. Then we have a table body where you have the whole, your data is there in the whole table. You can see the data here, all the data, whatever you should do. And last thing is something called footnotes. So the footnotes you can put two things, the abbreviations used in the table. So here you can see that avascular necrosis, AVN means avascular necrosis, CSLE means childhood SLE, not available, NA means not applicable, not available. So you have to put all the abbreviations used in the tables, and you also can say anything else is significant. Okay, so here we put one star mark. All comparisons with the AVN cases and controls were not significant. So mean follow-up age, which is not significant. Okay, so you can tell all those things in the footnotes. So any table should have 4 things. One is a table legend, one is a table column title, body of the table, and the footnotes. So types of tables, the tables are used in multiple ways. You can show the text instead of writing in this multiple sites of AVN. 2 of them had in shoulder, 1 of them had in the elbow, 8 of them had in the hip. Instead of writing like a big text, we can show it in a very simply in a table. And you also can show the statistical values in a table. And remember that whenever you make a table, I'll show you some examples. Ideally, so here this is a very bad, same table as put it in the bad example where you don't have any rounding of the decimals and proper alignment. So you should have a proper rounding of the decimals and you should align it properly so that the table looks very nice. You should not put a table with multiple colors and all those things should not be there. So center it, all of them should be centered, and you also should have rounding of the decimals. One, you should not have two decimals, 1 or 3 decimals like that. You should not have. So make it uniform. So the best practice is a contrast. Suppose if you are going to show 2 things in a table, so you can give one contrast. Heading can be in contrast. And the alignment is very, very important, because if you don't align, it won't look nice. And you can group the items similar into the one table. And spacing, remember that the table should have space so that the letters and all are very, very clear. When you come to the figures, so what is the difference? So tables will show what are the values you have. Figures, especially graphs and charts, show the changes over time. How does one related to the another? One parameter related to another parameter. How does this variable evolve over time? We can see the frequency distribution, and we can use it is used for correlation. Also, we can use. I'll show you examples for each and everything. So whenever I say the word figure, it could be a line graph, it could be a bar graph, it could be a pie diagram, it can be a drawings, flow chart, photographs, illustrations, maps. There are various types of figures are available. So graph shows the trends or patterns of the relationship. This is what very, very important. When you use a graph, it should show some trend or patterns of the relationship between the 2 variables. So that is what very, very important. So when you use a graph, we can use a single graph, bar graph, pie graph, flow charts. So all those things can be used. So what is a single graph? I'll show you a small example of a single graph. So here what they're trying to show, HIV prevalence in the countries in the last 10 years. What is the HIV prevalence among the pregnant women? So here what is a single line? So here the y-axis always represents the frequency. The x-axis represents the time. Okay. This is what you should, and always remember, y-axis you should start with 0. You should not start with 40, 50 and go up. And you determine the range. You want to make it big or small, determine the range of the values needed, and select the interval size also. Here every 1 year, every 2 years, every 3 years, whichever you want to show. So this is a single line graph. Okay. So what is a polygraph? So in the polygraph means 2 or 3 simple single line graph in the same graph. So here what you see here is, here they have shown the range for sex ratio in every decade in UP, Haryana, and in Delhi. So they have shown in 3 states what are the relationship between how does the sex ratio differs various years. So here is another single graph. So this is from the one study. What is the response of a drug given to children with arthritis? So the blue color is 30% response. The 50% response is red color. So you can do this also. This is also called a polygraph. That means multiple single line graph in one graph we can show. So this is called polygraph. Bar graphs are columnar diagram. This we use a lot of times. We use it as a simple bar diagram, compound bar diagram, or polybar diagram. I will show you each and everything. Again, remember, y-axis represents frequency, x-axis usually represents the time. So here is a bar diagram. So here the bar diagram shows in each month what is the rainfall in centimeters. So again here, from January to December, you can see that in July, there is a maximum rainfall is seen in July. So this is what you use a simple bar diagram. Clustered bar diagram means, so here, this is again the diagram from our paper. So here we have 2 groups of children. I told you, in all the children has SLE, one group has avian, another group has got no avascular necrosis. What is the difference in each clinical parameter? Suppose malar rash is more in non-avian. ANA, more positive in non-avian group. So you can show this, compare the values across the different categories. So first category is avian, the second category is non-avian, and you can show the different values across the categories. So this is called clustered bar chart. Compound bar chart. So what does this mean? So you can show in one bar, different things can be shown in a single bar. So different variables are shown in the, so here in each year, 2001, 2002, each year, what is the different types of power, electricity made from thermal, hydro, and nuclear power. So how much was taken? So in a single bar, we can show 3 different variables and this is called compound bar diagram. Okay. Stacked bar diagram is also a compound bar diagram. You do it in a stacked bar chart. So this all automatically comes in your PowerPoint. Okay, I'll show you that in the end. How does you do the PowerPoint? How we do these different types of chart? You just enter the data and you see what type of chart you want. It will show you all the different types of chart. Okay, so what is called polybar? Polybar diagram is a single line chart, and bar diagrams, bar chart also, both can be used in a single thing. So we can use like a multiple things. So here is a line and the bar graphs are drawn separately, or you can be shown in the same thing also. You can show this. So basically this one, what they're trying to show is the different timings, the methylene blue level. So they are showing it in both. So what happens in the BP and the lactate level and the methemoglobin level, and in every hour what is happening. So this is something called polybar diagram. Okay. So when you make the bar chart, make sure you make it in a good and clear way. I'm going to show you some bad examples so that you will, when you write your thesis, you won't do this. So this chart on the left side is a very bad example. You see multiple colors, multiple lines, and you have all the data. The same thing I have put it here as a good example. So this is very easier to read. Minimal support of components ensure data take a sense. So your data comes out very pop-up very nicely compared to this chart. Again, here the fertility rate in 2006 in different countries. And remember that if you put it in ascending order, you will know that it's very easy. So this is just you put it each and everything next to next. But if you do it in the ascending order, when you enter the data for making the chart, if you do it, when you enter it in the ascending way or descending way, it looks nice. And remember, don't make different colors. The colors, what you see in the computer screen will not be there when you take a printout of your thesis. So it will be completely distorted. Again here, so different colors. So don't try to do the different colors, just make it one color, and all the bars should be on equal size. One should not be thick, one should not be thin. So don't make like that. Okay, so something called scattered plots. Scattered plots usually shows relationship between the two variables. Okay, both are two different variables, but those shows the relationship. So here is basically a joint count done by a rheumatologist and done by the nurse. So whether it is correlating or not correlating. So to show the correlations, we use something called scattered plot. This is something called box and whiskers plot or a box plot. So this is one example I have shown in the right side. So here what we did is we estimated something called, there is something called PMP, platelet microparticles. What we saw in children with arthritis who have active disease. The red one is active disease. This is the inactive disease. You can see there's a mean value of the platelet microparticles are very high in children with active disease compared to the children who does not have active disease. So mean value is very high. So what does it mean? So here you have the minimum value. This is a maximum value. This first line is a 25th quadrangle range. This is a 75th quadrangle range. The center line is your median percentile. So this is something called box and whiskers plot. Okay, pie charts, you all know that is a 360-degree graphic presentation to show that different subclasses or categories based on your whole, how much is this category, how much is that category, this type, subtype, all those can be seen. Remember that again, don't try to make like this type of black 3D pictures and I'll make it very simple. 3D picture sometimes is not good when you put it in the paper, when you print it. So 2D is much better actually. That is regarding the pie chart. Histogram is used to show the distribution of the data. So how is the data is distributed? So that is very important. This is a frequency histogram to see whether the data comes under the typical bell-shaped curve or not. So this is your histogram. Correlation curves. Again, I already showed you correlation curves. By looking into the curve, you can see that how does this the correlation, whether it's a positive correlation or no correlation or negative or indirect correlation. So all those things can be seen in a correlation curve. Okay. There is something called area map. So what is this called area map? So this one, if you have like the prevalence of adults in Africa, so they show the different prevalence. So here, this is another, you will be seen in a lot of articles. Okay, it may not be useful for your thesis, but if you have a different places, I want to show the prevalence of each disease in each places. Where suppose if you take in CMC, we see a lot of people coming from all over India. You can put the India map and show the distribution of patients from each state is this much. We got 25 patients from West Bengal, 10 patients from Kerala. So we can show that also in this. So when you make a figure, the image clarity, the image should be very clear. And remember that the figure should be very clear and use a standard format and use legends to explain the key message. The figure, the very, very important thing is the legends, what you're writing, label all the important parts and give very specific things in the figures. So when you try to make the graphs, remember that now if you want to show it big, you can show this. So see here, overall trend of stability of the growth. You can see the growth is almost in the 100 only. But I want to show it very big. So here what happens, you start with 0, 50 and 100. You are starting with 100 and you're making the interval very small so that the difference looks like very huge. Don't do this. So whatever is there. So try to show that even though there is no difference, but by putting it in the graphical way, we are trying to show there is a huge difference, which is not ideal way of doing. Same thing here. Okay, so this one shows preoperative and postoperative. This is the difference between the treatment and the placebo group. But actually, when you see this, when you increase the difference here, you see here we're starting with 1 and going to 1.24. But here you see there is so much difference in the treatment group and the placebo group, pre and post-op, but actually there is no difference. But trying to split the numbers from 1 to 1.2, you are trying to do this, which is not a good way of doing. Same thing, the numbers, don't put same one, don't put a pie diagram, don't put a stacked bar diagram and a table. For a simple thing, either use any of one which is which you think it is relevant. Don't use all the three for this. This I already told you, 2D is much better than the 3D. Especially when it comes print also, the pie diagram, 2D is very, very good. And abbreviation. See, remember that don't write the word table to TAB. Figure you can write FIG dot you can write, but the table is not abbreviated. These are the international standards. When you write a manuscript, this is the way you have to write it. Okay. So the data representation strategies. So if you have a numerical data or a categorical data, distribution, example, frequency distribution, use the tables because this is only yes, no. So this side what numbers, that side what numbers. So when there is a numerical data, if you want to show some relationship, then you use the bar diagram. All those things can be used like histogram, dot plot, box and whiskers plot. When you have a numerical data, but you want to show between the two groups. When you have a categorical data, then you use bar chart, pie chart, pictogram. All those things can be used. Okay. So any questions you want me to answer? Okay. Yes. Okay. So all these graphs can be done by yourself in the PowerPoint. When you go to the PowerPoint.
Not to give another margin to the posterior. Okay.
Okay. So all those things can be shown in your PowerPoint. When you enter the data, it will show you what type of graph you want. It will show you the different types of graph. Okay. So next part of my talk is going to be on thesis writing parts and annexures. So when you make finally, how are you going to write? Okay. So how do you write your thesis? What are the annexures to be kept when you bind your thesis? I think this one already Dr. Tambuda told you about your MGR University requirement. So MGR University requirement, your thesis should be minimum of 75 pages, and not exceeding 100 pages of typed matter. This excludes your bibliography, all the other things like your annexures. So without the annexures, minimum should be 75 pages you should write and not exceeding 100, and it should be double line spacing on one side only, excluding certificates, acknowledgements, annexures, and bibliography. So your thesis material, your aims, introduction, aims, methodology, results, discussion, summary, recommendations. So this much alone should come around 75 pages minimum. And you should take 4 copies of your thesis and 2 copies on the CD, totally 6 to be submitted. Okay, before the prescribed date, the date may be changing this time because of our COVID. And how it is evaluated, you know that it will be sent to 4 different people, other head of the departments or units. So they will mark it either as approved or not approved. If it is not approved, you have to be resubmit the dissertation. If it is written, again, even whatever happens in the written or practical exam, the thesis no need to be done again. Okay. So what are the MD people doing is thesis, all the PhD people doing is dissertation. So that is totally different. The thesis means the way you do the thesis. We want to teach you when you're doing your MD thesis, MDMS, we want to teach you how to do a research project, not to find a big results, not a big, that is in your PhD. Okay, so PhD is called dissertation. MDMS people, what you're doing is thesis. Okay, so what are the key components when you write the thesis? The thesis should have a title page, acknowledgement, table of contents, should have abstract, introduction, literature review, methodology, results, discussion, conclusions, references and bar bibliography, and your appendices. And each part must be carefully thought and then you should write. Because remember that if you write your thesis very sincerely, it can be made into a manuscript very easily when you want to submit for a journal for publication. So I'm just going to show you an example for one of my PG who did a thesis on autoantibodies in pediatric SLE. So here in the first page, this is your thing, you should put the title, okay, and where it was done, okay, faculty and the department, place and date of completion. So second thing is your approval sheet, that is your certificate. Certificate should be signed by your PI, your guide, your head of the department, and the principal. So your supervisor, that's why they should do. So 3 things, your supervisor or your guide, HOD, and your principal. So all the 3 people has to sign this. So this is a typical format, you have to do it. And you can write the acknowledgement. You want to thank your guide, your wife, your God, everybody you can thank in the acknowledgement session. Then table of contents. So this is another important thing. So you should put the table of contents correctly aligned and you write what is there in which page each one starts. Then abbreviations. What are the abbreviations you used in the thesis? So you have to have a page for abbreviations. So this is basically a study on autoantibodies and SLE. So my PG has put all the abbreviations, whatever he is using in the thesis. And he also has put a list of tables and list of figures and which page it appears. Okay, so he has put so many figures. So he has put the page number for each figures. And he also put the tables. So which table is there in the which page. So this also has to be there. This all before starting your aims. Then the abstract. And the abstract, you should write it in the typical format like background, objectives, methods, results, and conclusion. So remember that abstract should not go more than 2 pages. So that is also very, very important. Then comes your introduction. So what comes in the introduction? So you introduce the topic to the person who is reading. So introduce the topic and you write what is the lacunae which is available. You can put references there and at the end of the introduction, you should write the hypothesis and what are you going to do? What is your hypothesis of the study and what are you trying to do? Okay, so this is what you should do. The last paragraph should contain either the aims or the problems that the study would want to achieve or solve. So that is your introduction. So introduce what is suppose SLE. So what is SLE? So what is the thing you are looking in the SLE patients and what are you trying to do by doing this study? Then the literature review, this should be extensive. And remember that it's not the number of pages, it's all should be most recent literature review. Because when you write this as a manuscript, the experienced person will find out why this person has not included this recent available literature. So you should be able to get all the recent literature, whichever is relevant to your study and make it right, start writing that. Okay, so I told you quality is more important than the quantity. And you should also remember that how it is going to impact your study. So what are you trying to do? Is it already done, not done? All those things should come under the literature review. Then when it comes to, so literature review also contains references. In the methodology, so what is your research design? Who is your inclusion criteria, exclusion criteria? That is your population and the samples, what you're going to use in the study. Then your research instrument. So what are the, suppose it's a new test. So what is the test you are doing? What are the scales you are using? So all has to be written in the methodology section, and how is the data gathered, okay, and how is the data processed, and what is the statistical treatment or statistical plan you made for the study. So all those things should come, and after this you should write about the IRB acceptance for this, who approved your thesis. So methodology is over. So methodology session will end with IRB clearance and your statistical methods. Then the results page. So results page only we are talking. In the results page, we are talking about how to present your results, either in the tabular form, either in the figure, or in the text form. So depending on what type of data you have, you start writing your results. Remember that in the results, don't make any conclusion. Okay, don't make any conclusion from the results thing, because we are going to make the conclusion in the discussion part. You have to just present. So here, this section must be mere a statement of observations and not interpretation of your results. Discussion part. So discussion part, in-depth exploration of the topic being studied, and you should compare your results with the previous studies done. That person has done the study and find out this is what happened. So now this my study did not was comparable with that study, or we found it new. So we have to discuss like that. Each and every positive and negative results of your study should be discussed with the existing literature. Okay. So then what is at the end of the discussion, you should write what are the limitations of the study. So what is your study missing? So that you have to find out. Summarize. So salient findings and results of the hypothesis tested. So what do you find out at the end of the day? And conclusion, what are the key messages, what you find out, and what is your results implicate. So by this study done, this has shown, this has been done in a larger cohort of patients to see more effects. So that's what you have to conclude. Recommendations. Is there any recommendations from your study? Based on this study, we are, the treatment has to change. We have to do this, that we can write as a recommendation. References versus bibliography, very, very important thing. So what we write in thesis is called references. References means whatever you have read and you cited in your work. Bibliography means whatever you read for your study. Bibliography may be 100, but remember that we cannot put all the 100 articles in your thing. So reference means whatever you quoted in the introduction page, or in the literature review, or in the discussion part. Okay, results part should not have any references. Okay, and most of the time, Vancouver Style is the one approved for any standard article. So try to write all the things in the Vancouver Style. All your references should be in a Vancouver Style. This is all available in all the standard books. And the appendix. So what will you keep at the end? So your case report form, your questionnaire, your pro forma, consent form, IRB certificate, IRB approval, the original master sheet of the Excel, the original scales. And if you're doing from outside, you did the study, any government authority, you got permission from outside. Okay. Formatting is very, very important. You should use the same font throughout your thing. You should not keep on changing the fonts. Ideal fonts will be either Arial or.
What I understood is there or not.
Okay. The other important thing is plagiarism. This is what very, very important. So plagiarism, there is a tool by the MGR University website. You have to upload that and you have to get a certificate. Remember that up to 25% of similarity can be there in your thing. More than that, it will say if it is more than 25% similarity, it is called plagiarism. So at the end of the study, you have to get a plagiarism report from the website, and it has to be signed by your supervisor. So summary, organization of the material is a very key. Good writing. So learning is key. Start writing. Pay attention to each and every details. Show it to your guide. Go for multiple corrections has to be done before it gets into a full shape. Okay. So with this, I am going to stop it here. I am happy to answer any questions.